Chapter 13 Photosynthesis in Higher Plants. All animals, including amphibians, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all the other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light and energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. That is, it is the primary source of all the food on Earth. It is also responsible for the release of oxygen into the atmosphere for green plants. Have you ever heard that what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, grey pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in green plants of the leaves in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2, while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two plants of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while that portion that was in the tube tested negative. This shows that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiments. It is interesting to learn about those simple experiments that led to the gradual development in the understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770, performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in the 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon extinguished. Figure 13.1 A, B, C, and D. Figure 13.1 Priestley's experiments A, B, C, D. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Ingenhouse, 1730 to 1799, showed that sunlight is essential to the plant process that somehow purifies the air found by burning candles of breathing animals. Ingenhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight small bubbles were formed around the green parts, while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence of production of glucose when plants grow. Glucose is usually stored as the starches. Later studies showed that the green substance in plants, chlorophyll as we know it now, is located in special bodies later called chloroplasts within the plant cells. He found that the green parts in plants is where glucose is made, and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann, 1843 to 1909. Using a prism, he split light into its spectral components and then illuminated a green alga. Cradophora placed in a suspension of aerobic bacteria. The bacteria were used to detect the signs of photoevolution. He observed that the bacteria accumulated mainly in the region of the blue and the red light of the split spectrum. First, action spectrum of photosynthesis was just described. It resembles roughly the absorption spectra of chlorophyll A and B discussed in section 13.4. By the middle of the 19th century, the key features of a plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as CO2 plus H2O in the process of light gives CH2O plus O2, where CH2O represents a carbohydrate, for example glucose is 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by microbiologist Cornelius Van Neal in 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidized compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 in the presence of light gives 2A plus CH2O plus H2O. In green plants, H2O is hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis, when H2S instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that O2 evolved by green plants comes from H2O and not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O in the presence of light gives C6H12O6 plus 6H2O plus 6O2. Where C6H12O6 represents glucose. The O2 released is from water. This was proved using radioisotopic techniques. Noted that this is not a single reaction but the structure of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as a substrate are used in the equation given below? 13.3. Where does photosynthesis takes place? You would of course answer in the green leaf or in the chloroplast. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a larger number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimal quantity of the incident light. When do you think the chloroplast will be aligned with their flatter surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast there is membranous system consisting of grana, the stroma lamellae and the matrix stroma. Figure 13.2. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar which in turn forms a starch. The former set of reactions, since they are directly light driven, are called light reactions. Photochemical reactions, the latter, are not directly light driven but are dependent on the products of light reactions ATP and NADPH. And to distinguish the latter, they are called by the convention as dark reactions. Carbon reactions, however, this should not be construed to mean that they occur in darkness or that they are not light dependent. Figure 13.2. Diagrammatic representation of an electron micrograph of a section of chloroplast. Outer membrane, inner membrane, stroma lamellae, grana, stroma, ribosome, starch, granule, lipid droplet. (13.4) How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? We can look for an answer to this question by trying to separate their leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to the single pigment but due to 4 pigments. Chlorophyll A, bright or blue-green in the chromatogram. Chlorophyll B, yellow-green, xanthophylls, yellow, and carotenoids, yellow to yellow-orange. Let us now see what role various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us now study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelengths. Figure 13.3a. Graph showing the absorption spectrum of chlorophyll A, B and carotenoids. Absorbance of light by chloroplast pigments. Chlorophyll A, chlorophyll B, carotenoids. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the big yaw. From figure 13.3a, can you determine the wavelength color of the light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at figure 13.3b, graph showing action spectrum of photosynthesis. Rate of photosynthesis measured by oxygen, that is photo release. Showing the wavelengths at which maximum photosynthesis occur in a plant. Can you see that the wavelengths at which there is maximum absorption of chlorophyll A, that is in the blue and the red region, also shows a higher rate of photosynthesis? Hence we can conclude that chlorophyll A is a chief pigment associated with the photosynthesis. But by looking at figure 13.3c, graph showing action spectrum of photosynthesis superimposed on absorption spectrum of chlorophyll A. Light absorbed, rate of photosynthesis, absorption. Can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and the red regions of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelengths of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. 13.5. What is light reaction? Light reactions or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates. ATP and NADPH, several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, LHC, within the photosystem 1 PS1 and photosystem 2 PS2. These are named in the sequence of the discovery and not in the sequence in which they function during the light reaction. LHC are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system, also called the antenna. These pigments help to maintain photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer. Hence is called P700, which while in PS2 it has absorption maxima at 680 nanometer and is called P680. Figure 13.4, light harvesting complex, the photon, reaction center, pigment molecules, primary acceptor. 13.6, electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into the orbit farther from the atomic nucleus. These nucleus are picked up by an electron acceptor which passes them to an electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem PS1 simultaneously. Electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADP+. The addition of these electrons produces NADP+ to NADPH+H+. This whole scheme of transfer of electrons starting from the PS2 up into the acceptor down the electron transport chain PS1, excitation of electrons transfer to another acceptor and finally downhill to NADP+, reducing it to NADPH+H+ is called a Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. Figure 13.5 Z scheme of light reaction. Photosystem 2, photosystem 1. Light, let's see, electron from acceptor, electron transport system to let's see that. And then let's see 2, electron acceptor in PS1, to P2, UDP plus 2. UDPH. Let's see of PS2, H2 against 2 electron plus 2 H plus. 13.6.1 Splitting of water. You would then ask, how does PS2 supply electrons continuously? Electrons that were moved from PS2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with PS2 water is split into 2H plus nascent oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. Electrons needed to replace these removed from photosynthesis 1 are provided by photosystem 2. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. 2H2O gives 4H plus O2 plus 4 electrons. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed, likely to be released in the lumen or on the outer side of the membrane? 13.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP, an inorganic phosphate, in the presence of light. When the two photosystems work in a series, first PS2 and then PS1 are a process called non-cyclic photophosphorylation occurs. The two photosystems are connected to an electron transport chain as seen earlier in the Z scheme. Both ATP and ADP H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamellae. While the membrane or lamellae of the grana have both a PS1 and PS2, the stroma lamellae membranes lack a PS2 as well as an ADP reductase enzyme. The excited electron does not pass on to the NADP plus but is cycled back to the PS1 complex through the electron transport chain. A cyclic flow hence results only in the synthesis of ATP but not of ADP H plus H plus. Cyclic photophosphorylation also occurs when only light of wavelengths beyond the 680 nanometer are available for excitation. Figure 13.6 Cyclic photophosphorylation. Light to chlorophyll P700, ADP plus IP to ATP electron transfer system to chlorophyll P700, chlorophyll P700 to electron acceptor PS1, electron acceptor to ATS, electron transport system. 13.6.3 Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism like respiration and photosynthesis too. ATP synthesis is linked to a development of a proton gradient across the membrane. Just as these are the membranes of thylakoid, there is one difference though. Here, the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, proton accumulate in the intermembrane space of the mitochondria when electrons move through the ETS, chapter 14. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons and their transport to determine the steps that cause the proton gradient to develop. A. Since splitting of the water molecules takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the membrane of the thylakoids. B. As electrons move through photosystems, protons are transported across the membrane. This happens because the primary acceptor of the electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes the proton from the stroma by transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane, along with electrons that come from the acceptor of the electrons of PS1. protons are necessary for the reduction of NADP plus to NADP H plus H plus. Protons are necessary for the reduction of NADP plus to NADP H plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in the number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in the pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF not of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF naught is embedded in the thylakoid membrane and forms a transmembrane channel that carries a facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy back to ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. Energy is used to pump the protons across the membrane to create gradient for a high concentration of protons within the thylakoid lumen. ATP synthase is a channel that allows the diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reactions taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. Figure 13.7 ATP synthesis through chemiosmosis. Stroma NOH plus light P6ATPase2 complex plastoquinone H2 H plus ions PQ2 PQH2 H plus is given cytochrome P6F Plastocyanine PC and P700PS1 complex light energy smile Plastocyanine PC2FD into FD2FNR NADP plus H plus to NADPH lumen high H plus concentration H plus from P6ATPase2 results and plastoquinone H plus ATP synthase CF naught complex CF1 ATP synthase ADP plus IP gives ATP. From ATP synthesis presents out high electrochemical potential gradient, low stroma level, thylakoid membrane. 13.7 Where are the ATP and NADPH used? We learned that the products of the lighter reaction are ATP, NADPH, O2 and O2. Of these, O2 diffuses out of the chloroplast, while ATP and NADPH are used to drive the processes leading to the synthesis of food. More accurately, sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but is dependent on the products of the light reaction, that is ATP and NADPH. Besides the CO2 and H2O, you may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again and then stops. If then light is made available, the synthesis starts again. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourself. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2 to produce a CH2O whole n times of sugars. It was of interest to scientists to find out how this reaction proceeded, or rather, what was the first product formed when CO2 is taken into the reaction or fixed just after boiling molecule. Among the several efforts to go to radioisotopes to beneficial use of Melvin Calvin study and is exemplary. The use of a reductor C14 by him in algal photosynthesis. These led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working on the complete biosynthetic pathway, hence it was called as Calvin cycle. It was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants, where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types. Those plants in which the first product of CO2 fixation is a C3 acid, that is PGA, that is the C3 pathway, and those in which the first product was a C4 acid, OAA, which is the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. 13.7.1 The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting fixing CO2, would have 3 carbons of PGA? The study is very unexpectedly showed that the acceptor molecule was a 5-carbon ketose sugar ribulose phosphate RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2-carbon compound. They spent many years trying to identify 2-carbon compound before they discovered the 5-carbon RUBP. 13.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathways. For ease of understanding, the Calvin cycle can be described under the 3 stages: carboxylation, reduction, and regeneration. First, carboxylation. Carboxylation is the fixation of CO2 into stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the ATP for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or RUBisco. Second, reduction. These are a series of reactions that led to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of ADP for reduction of CO2 molecule. Fixation of 6 molecules of CO2 in 6 rounds of the cycle are required for the formation of 1 molecule of glucose from the pathway. Third, regeneration. Regeneration of the CO2 acceptor molecule, RUBP's portion of the cycle, is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. And for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make out 1 molecule of glucose, 6 turns of cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. It might help you understand all of this if you look at what goes in and what comes out of the Calvin cycle. In 6 CO2, out 1 glucose, in 18 ATP, out 18 ADP, in 12 NADPH, out 12 NADP. Figure 13.8 The Calvin cycle proceeds in stages. First, carboxylation, during which CO2 combines with ribulose 1,5 bisphosphate. Second, reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH. And third, regeneration, during which the CO2 acceptor ribulose 1,5 bisphosphate is formed again, so that the cycle continues. Ribulose 1,5 bisphosphate from atmosphere CO2 plus H2 carboxylation. First process gives 3-phosphoglycerate. ATP plus NADPH gives ADP plus IP plus NADP plus. Second reaction gives a triose phosphate, which is sucrose and starch. and in the present region is IDP and ADP. And in the presence of regeneration and ATP and ADP, this is where the ribosome 1,5 is phosphate. .8 the C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid is as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they different from the C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate high temperature. They show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical sections of leaves, one of a C3 plant and the other of a C4 plant. Do you notice the difference? Do both have the same type of mesophyls? Do they have a similar cells around the vascular bundle sheath? Particularly large cells called the vascular bundles of the C4 plants are called bundle sheath cells. And the leaves which have such anatomy are said to have crans anatomy. Crans means a wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having large number of chloroplasts, thick walls impervious to gaseous exchange and no intracellular spaces. You may like to cut a section of the leaves of C4 plants, maize or sorghum to observe the crans anatomy and the distribution of the mesophil cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope. Look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you identify the C4 plants. Now study the pathway shown in figure 13.9. This pathway that has been named the hatch and slack pathways again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 accepts the risk of 3-carbon molecule phosphonylpyruvate PEP and is present in the mesophil cells. The enzyme responsible for the fixation is PEP carboxylase of PEPcase. It is important to register that the mesophil cells like a Rubisco enzyme, the C4 acid OAA is found in the mesophil cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophil cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release a CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophil where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase Rubisco but lack PEPcase. Thus, the basic pathway that results in the formation of the sugars, the Calvin pathway, is common to the C3 and the C4 plants. Did you note that the Calvin pathway occurs in all the mesophil cells of the C3 plants? In the C4 plants, it does not take place in the mesophil cells, but does so only in the bundle sheath cells. 13.9 Diagrammatic representation of the hatch and slack pathway. Mesophil cell, atmospheric CO2, plasma, membrane, cell wall, HCO3 minus phosphonylpyruvate. Fixation, C4 acid transport, plasma does not have bundle sheath cells, C4 acid decarboxylation, CO2 outside fixation by Calvin cycle, C3 acid transport, which gives C3 acid universal regeneration used by phosphoenolpyruvate. Mesophil cells, bundle sheath cells. Content 1.9 Photorespiration. Let us try and understand one of the processes that creates an important difference between C3 and C4 plants, photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA. That is catalyzed by Rubisco. RUBP plus CO2 in the presence of Rubisco gives 2 times of 3-PGA. Rubisco, that is the most abundant enzyme in the world. Do you wonder why? Is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2 when the CO2 is to O2 is really equal than for O2. Imagine what would happen if this were not so. This binding is comparative. It is a relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some O2 does bind to rubisco and hence CO2 fixation is decreased. Here, the REBP instead of being converted to two molecules of PGA binds with O2 to form one molecule of phosphoglycerate and phosphoglycolate to carbon in the pathway of photorespiration. In a photorespiratory pathway there is neither synthesis of sugar nor of ATP. Rather, it results in the release of CO2 with utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not yet known. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release CO2. This results in increasing the intracellular concentration of CO2. In turn, this ensures that the rubisco functions as carboxyl, minimizing the oxygenase activity. Now that you know that C4 plants lack of photorespiration, you probably can understand why the productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperatures. 13.10 Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of the plants, including crop plants. Photosynthesis is under the influence of the several factors, both internal plant and external. The plant factors include the number, size, age, and orientation of leaves, mesophyll cells, and chloroplast, internal CO2 concentration, and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of the sunlight, temperatures, the CO2 concentration, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, though several factors interact and simultaneously affect the photosynthesis or CO2 fixation, usually one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factors available at suboptimal levels. When several factors affect any biochemical processes, Blackman's 1905 law of limiting factors comes into effect. This states the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of a green leaf and optimal light and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. 13.10.1 Light. We need to distinguish between light quality, light intensity, and the duration of the exposure to light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between incident light and CO2 fixation rates at low light intensities. At higher light intensities, gradually the rate does not show further increase as other factors become limiting. What is interesting to note is that the light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or intense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. Figure 13.10 Graph of light intensity over rate of the photosynthesis. Rate of photosynthesis, light intensity A, B, C, D, 13.10.2 Carbon dioxide concentration. Carbon dioxide is a major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over longer periods. The C3 and C4 plants respond differently to CO2 concentrations. At low light conditions, neither group responds to high CO2 conditions. At high light intensities, both the C3 and C4 plants show increase in the rates of photosynthesis. What is important to note is that the C4 plant shows saturation to about 360 micro show that the saturation of 360 micro L per liter, while C3 responds to increase CO2 concentration and saturation is seen only beyond 450 micro L per liter. Thus, current availability of CO2 levels is limiting to C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rates of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide and enrich the atmosphere that leads to higher yields. 13.10.3 Temperature. The adaptation being enzymatic are temperature control. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plants respond to higher temperatures and show higher rate of photosynthesis, while C3 plants have much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. 13.10.4 Water. Even the water is one of the reactants in the light reactions. The effect of water as a factor is more through its effect on the plant rather than directly on the photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes the leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Summary: Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by the leaves, stomata, and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly the leaves. Within the leaves, the mesophyll cells have a larger number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are site for the light reactions, while the chemosynthetic pathways occur in the stroma. Photosynthesis has two stages: the light reactions and the carbon-fixing reactions. In the light reactions, the light energy is absorbed by the pigments present in the antenna and funneled to the special chlorophyll A molecules called reaction center chlorophylls. There are two photosynthesis systems, PS1 and PS2. PS1 is a 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has a P680 reaction center that absorbs red light at a 680 nanometer. After absorbing light, electrons are excited and transferred through PS2 and PS1 and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the protons gradient due to the movement through the F0 part of the ATP synthase enzyme releases enough energy for the synthesis of ATP. Splitting of water molecules is associated with the PS2, resulting in the release of O2 protons and transfer of electrons to PS2. In the carbon fixation cycle, CO2 is added to the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by the Calvin cycle, and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes the basal oxygenation reaction in C3 plants photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of the CO2 fixation takes place in the mesophyll as a 4-carbon compound in the bundle sheath cells. The Calvin pathway is carried out for the synthesis of carbohydrates.